name is Andrew Gillis. I'm a Royal Society University Research Fellow in the Department of Zoology at the University of Cambridge and a longtime summer investigator at the Marine Biological Laboratory in Woods Hole. I'm also really excited to be joining the faculty of the MBL as an associate scientist next year. My lab studies the embryonic development of the skate, but before I tell you a little bit about our work and our favorite animal, I'd like to start by just showing you a couple of short movies. The first movie that I'd like to show you is of a lamprey. So a lamprey is a type of fish with a long eel-like body, and in this video you can see the lamprey swimming through the water with an undulating motion, seeking out a larger fish that it can attach to. So lampreys are parasites, and they latch onto passing fishes with their specialized sucker organ, and then once attached, they rasp a hole in the side of the fish's body, and they feed on that fish's tissues and bodily fluids. So now we can contrast the lamprey with another type of fish, a great white shark. And in this amazing footage, we can see a great white shark preying on a seal. So the white shark propels its massive body toward the surface of the water at great speed, and then the shark actually breaches the water as it grabs a seal and then comes crashing back down again. We might ask ourselves, what are the anatomical differences that allow us to distinguish a shark from a lamprey? And in this case, I'm not just talking about differences in body size, but rather are there specific body parts that are present in a shark but absent in a lamprey, which contribute to the striking differences in lifestyle and behavior that are exhibited by these two very different fishes. So to find the answer to this question, it might be helpful if we take a look at the skeleton beneath the skin. So if we compare the skeleton of a lamprey with the skeleton of a shark, we can actually observe quite a few differences, but two in particular might jump out at you. So first we can see that the shark has biting jaws, while the lamprey doesn't. So the lamprey does have a mouth with a supporting skeleton, but in the lamprey this skeleton doesn't take the form of biting jaws. And second, we can see that the shark has paired fins, one pair near the head called the pectoral fins, and one pair further down the body called pelvic fins. The lamprey, on the other hand, doesn't have these paired fins. So if we take a look at the human skeleton, we can see that we also have biting jaws and paired appendages, much like a shark. Although, of course, our paired appendages take the form of limbs rather than fins. But like in the shark, we have one pair of limbs near our head, and this is our shoulders and our arms. And we have another pair of limbs further down our body, and this is our pelvis and our legs. The reason that sharks and humans share jaws and paired appendages is because both groups have inherited these structures from a common ancestor that lived hundreds of millions of years ago. So both sharks and humans belong to a group of animals called the jawed vertebrates, and my lab is interested in reconstructing the early evolutionary history of jawed vertebrates by studying embryology and the molecular and cellular mechanisms that jawed vertebrates use to build things like jaws and fins and limbs. So by comparing the development of the skeleton across jawed vertebrates, we're able to discover shared ancestral features that were present at the origin of this group, and we're also able to discover unique features that have evolved during the independent evolutionary history of particular lineages. So we currently know a great deal about the development of the skeleton in bony vertebrates, and this comes largely from studies in animals like the mouse and the chick and the frog and the zebrafish. But we know comparatively little about the development of the skeleton in cartilaginous fishes, which is the group of animals that includes the sharks and the skates and the stingrays. And without this information, we are unable to understand the very earliest stages of jawed vertebrate evolution. And so for much of the past 10 years, my lab and I have focused on studying the development of the skeleton in cartilaginous fishes. And we focused on one cartilaginous fish in particular, and that is the skate. If you spent any time on the east coast of North America, you may have come across skates washed up on the beach, or perhaps it's more likely that you've come across washed up skate egg cases which look something like this, and which are often called mermaid's purses or devil's purses. So most skates are relatively small cartilaginous fishes, and this, combined with their benthic lifestyle, means that it's relatively easy to maintain these animals in an aquatic facility like the Marine Resources Center at the MBL. And because skates are oviparous, or egg-laying, it means that they are particularly well suited for embryological study, because we can collect the eggs as they're laid in a tank and then study the development of the embryos inside the egg case. The species of skate that we study at the MBL is called the little skate, Leucorrhea arenacea. And in the following video, I'll give you a brief overview of the embryonic development of the little skate. So skate eggs are a lot like chicken eggs in that they generally contain a single ball of yolk with a single embryo developing on top. So if we take a look inside the egg just after it's laid, at the earliest stages of embryonic development, the embryo is recognizable as a simple disc of cells sitting on top of the ball of yolk. But soon enough, we begin to see a thickening along the edge of that disc, and that thickening will elongate as the embryo forms and grows. So already at these stages, we can see a head end of the embryo, which in the video is pointing toward the top of the screen, and a tail end of the embryo, which is pointing toward the bottom of the screen. And once the embryo forms a distinct head and tail, it begins to wiggle. So these embryos will continue to elongate inside the egg case for several weeks, at which point we can actually remove the embryo from the egg case and observe it in a dish of seawater. Now, at these later stages of development, we begin to see the appearance of some features that are recognizable in the adult skate. So things like early forming gills and fin buds. So I mentioned that we typically see a single embryo developing on a ball of yolk, but very occasionally we do come across twins where we can actually see two embryos developing on the same ball of yolk. So once the embryos have grown to fill the inside of the egg case, they're ready to hatch. And the embryos hatch by pushing with their tail against the back of the egg case, and then they emerge headfirst out of the opposite end of the egg case. And when they emerge, they look like miniature adults, and they're basically ready to go off and live independently, feeding on the seafloor like adult skates do. So because we can open and observe and manipulate skate embryos, we can actually use these as a model system to understand the development of the skeleton in cartilaginous fishes. So for example, we can open the early embryo and label specific cells and then follow their movements within the developing egg. Or we can turn particular genes on or off at different times of development and then look at the effect that this has on the development of the skeleton. So what I'm going to do now is give you an example of some of the work that we're doing to look at mechanisms of skeletal development in the little skate. And in particular, I'm going to tell you about a project that we're working on that aims to try and resolve the evolutionary origin of one of those key jawed vertebrate anatomical features, paired appendages. Nearly 150 years ago, the comparative anatomist Carl Gegenbauer proposed a theory on the evolutionary origin of paired fins. So Gegenbauer was studying skeletal anatomy, and he noticed some striking parallels between the skeleton of the fins and the skeleton of the gills. So if we take a look at this drawing of a shark skeleton, and if we look at the fin region, you can see that there's a bar of cartilage located close to the body, and this is called the pectoral girdle, and I've colored it in yellow. And you can see the fin skeleton itself, which articulates with that girdle and then projects off the side of the animal, and I've colored that in red. So if we move forward and we take a look at the organization of the gill skeleton, we can see that the gills are supported by a bar of cartilage that's located close to the side of the head. This is called a gill arch, and I've colored those in yellow. And by a series of cartilaginous rods called branchial rays that project off the gill arch, and I've colored those in red. So Gegenbauer noticed parallels between the organization of the fin skeleton and the girdle, and the branchial rays and the gill arches, and he proposed that paired fins evolved by transformation of a gill arch. So for a long time, this theory has been cast aside as fundamentally flawed and really as more of a historical curiosity. And this is partly because there's no fossil evidence documenting this transformation from a gill arch into a fin, but also because there was no developmental evidence of similarities between developing gill arch skeletons and developing fin skeletons. So for example, there was no evidence of common molecular mechanisms controlling the development of these skeletal features, and it was widely thought that the gill skeleton and the fin skeleton arose from different cell populations within the embryo. Gegenbauer's theory of fin evolution by transformation of a gill arch was based largely on the skeletal anatomy of cartilaginous fishes, sharks and skates. And so when we began to work on the embryonic development of the skate, we became interested in revisiting and reevaluating this classical theory. So in particular, we wanted to find out whether there might be previously unrecognized developmental parallels between the gill skeleton and the fin or limb skeleton of jawed vertebrates that are perhaps uniquely present in cartilaginous fishes. And so to do this, we started by looking at some of the mechanisms that are important for controlling the development of fins and limbs and testing whether these same mechanisms might be at play in the developing gill arches of the skate. So if we take a look at the development of the limb in a jawed vertebrate embryo, so for example, in this chicken embryo, we can see that the limb first appears during embryonic development as a bud that protrudes from the side of the body. And during the 1960s, embryologists did some beautiful experiments where they grafted tissue from the back of the limb bud of one embryo into the front of the limb bud of another embryo. And when they did this, they noticed something really striking. So the chicken limb skeleton normally has 3 digits. And when you take tissue from the back of the limb bud and transplant it to the front of the limb bud, what you find in that embryo is that they develop a limb with 6 digits. And not only that, but the 3 extra digits are oriented in a mirror image relative to the normal digits. It was later discovered that the factor that's expressed in the posterior limb bud that's responsible for this effect is a gene called sonic hedgehog. So the sonic hedgehog gene encodes a developmental signaling molecule that can influence the decisions that cells make within the limb bud. So what I'm showing you here is a picture of a chicken limb bud with expression of the sonic hedgehog gene in the posterior limb bud indicated by dark purple. So if you misexpress sonic hedgehog in the anterior limb bud, where it's not normally expressed, what we find is that we again get the same effect on the skeleton. So instead of forming the normal 3 digits, you form 6 digits, and the new digits are oriented in a mirror image relative to the normal digits. And conversely, if we look at a chicken limb bud that doesn't express sonic hedgehog, we see the opposite effect. So we see the reduction of the limb skeleton. So you can see in this case, the limb skeleton completely lacks digits. So we were interested in first testing whether or not the sonic hedgehog gene is active during the development of the gill arch skeleton in the skate, and whether it might be playing a similar role controlling the number and the orientation of skeletal elements within the gill arch. So first we looked at the expression of the sonic hedgehog gene in developing skate embryos, and we found that it is actually expressed in the developing gill arches. So in skates, the gill arches develop as bulges that sort of grow out from the side of the head. And we found that the sonic hedgehog gene is expressed in a stripe that runs down these expanding arches. And you can see it in this image here. Again, as in the previous slide, the sonic hedgehog gene expression is indicated with a dark purple staining. So we found that this gene is expressed in a specialized population of cells that we called the gill arch epithelial ridge, or the GAER. And it turns out that this cell population is really important for controlling the development of the skeleton in the gill arches. So we did some experiments where we would either overactivate the sonic hedgehog signaling pathway in the developing gill arches of a skate embryo, or inhibit the pathway in the developing gill arches. And we looked at the effect that that would have on the developing skeleton. So the way we would do this is we would take microscopic beads that were either loaded with sonic hedgehog protein to overactivate the pathway, or beads that were loaded with a drug called cyclopamine, which blocks the sonic hedgehog signaling pathway. And this would allow us to block that signal. And when we did this, we noticed something really striking in the gill arch skeleton. So first I'm going to show you a picture of a normal skate gill arch. And on the upper half of the gill arch, you can see that there are 5 of those little cartilaginous rods called branchial rays. And I've numbered those 1, 2, 3, 4, and 5. So if we implant a bead in the developing gill arch that's loaded with sonic hedgehog protein, we find that that's able to induce the formation of extrabranchial rays, which I've indicated with a little red asterisk. And not only are there extra rays, but those extra rays are actually organized in a mirror image orientation relative to the normal rays. So they originate on the opposite side of the arch, and they curve in the opposite direction of the normal rays. And conversely, if we block the sonic hedgehog signaling pathway by implanting beads loaded with that drug, cyclopamine, we find that the opposite effect is seen. So we see a reduction in the number of branchial rays on that arch. So in this case, we only see 3 branchial rays where there should normally be 5. And so these experiments point to a striking developmental parallel between developing fins and limbs and developing gill arches in the skate. So the sonic hedgehog gene in both of these structures seems to be important for controlling the number of skeletal elements that forms and the arrangement of those skeletal elements within the organ. So I told you that another outstanding problem with Gegenbauer's theory of gill arch origin of fins was the idea that gill arches and fins arose from different cell populations within the embryo. So classically, it was thought that the gill arch skeleton of fishes derived largely from a population of cells called neural crest cells. And if you look at this picture of an early skate embryo, you can see that I've colored in the developing central nervous system in blue. And the neural crest cells form within the developing central nervous system, but then crawl away and they migrate down and populate the head, where they will give rise to much of the skeleton of the face. So for example, the jaws and so on. Now the fin skeleton was thought to arise from a completely different population of cells called the lateral mesoderm, and I've colored that here in red. And so because these skeletal elements were thought to arise from different cell populations, it seemed inconceivable that one could have given rise to the other. So in order to test this in skate embryos, we developed methods for labeling cells within the early skate embryo so that we could follow where they end up in the skeleton, and then we could directly test which skeletal elements formed from the neural crest and which elements formed from lateral mesoderm. This process is called cell lineage tracing, and to begin with, we need to select embryos of the appropriate developmental stage for labeling. And so to do this, we use a bright flashlight to illuminate the egg so that we can see the embryo inside. And we select embryos of the appropriate developmental stage for our labeling experiments. So once we've selected our embryos, we can then cut a window in the top of the egg case to expose the embryo, and under the microscope, we can microinject either the neural crest or the lateral mesoderm with a fluorescent dye. We can then seal up the egg again with a piece of donor eggshell and some superglue, pop the egg back in a sea table of normal seawater, and then leave the egg to develop until the embryo has a skeleton. And once we hit this point, we can then cut thin sections through the embryonic skeleton and recover the cells that we labeled in the very early embryo. And this allows us to directly test which elements of the skeleton come from the neural crest and which elements of the skeleton come from the lateral mesoderm. I told you that the neural crest cells form from the developing central nervous system. And so to label these cells, we microinject the early embryonic brain region with a fluorescent dye called dye I. And if we look at these embryos 5 days later, we can already see neural crest cells crawling away from the developing central nervous system down into the embryonic head. Now, when we follow the fate of these cells into the skeleton, we find, as expected, that these neural crest cells contribute to the skeleton of the head, including things like the jaws and the gill arch cartilages. And in these images, I've outlined the early embryonic cartilages with a dashed line and a yellow coloration, and you can see the fluorescent pink cells that were labeled in the early embryo in these cartilaginous elements. Now, if we next label the lateral mesoderm in early skate embryos, we find, again not surprisingly, that we can follow these cells into the developing fin skeleton. But what was quite surprising is that in addition to the contribution to the fin skeleton, we found that the lateral mesoderm cells also contribute to the gill arch skeleton in the skate. So for example, in this image, you can see labeled cells from the lateral mesoderm that have found their way into the branchial ray of a gill and also into the gill arch cartilage itself. An additional experiment that we did was to label the neural crest and the lateral mesoderm simultaneously in the same embryo. So in this case, we labeled the neural crest with a pink fluorescent dye called dye I, and the lateral mesoderm with a green fluorescent dye called dye O. Now, when we grew these embryos up to the point where they had a skeleton, and we looked at the gill arches, we could see that the gill arches were actually made from both neural crest and lateral mesoderm-derived cells. So these experiments suggest that contrary to received wisdom, the gill arches and the paired fins don't actually arise from different cell populations within the embryo, but rather they arise from a common pool of cells that seems to form from a mixture of lateral mesoderm and neural crest around the boundary between the embryonic head and the embryonic trunk. So by studying the embryology of the skate, we were able to discover unrecognized developmental parallels between gills, fins, and limbs, which I think point to a deeper evolutionary relationship among these structures than was previously appreciated. And these developmental parallels might also account for the anatomical parallels that were noted by Carl Gegenbauer over a century ago. And I also hope that this work illustrates how venturing outside of the laboratory and exploring more of nature's incredible diversity can shed light on important basic mechanisms of animal development, and might also shed light on the evolutionary origin of our own body plan. So thank you very much for listening. I hope you've enjoyed this, and I'm very happy to answer any questions that you might have if you post them in the comment section below.